Okay, let's get started with our lecture. Um, so last time we were introducing how B cells make immunoglobulins, and we were talking about the process of VDJ recombination, which is a pretty complicated process and I think a very difficult concept to, I think, kind of get comfortable with. So I'm going to spend some more time talking about that today. And hopefully, I think all of you can at least appreciate the mechanism by which this takes place, because it's really critical for understanding how it can be that our body is able to produce such a tremendous diversity of different immunoglobulins that form the basis of things like antibodies. Let's start off first by checking in on where we're at in our semester so far. So here we are in week 4. So today we are going to talk about the third part of our discussion about antibodies and B cells. Then we are going to spend 3 lectures talking about T cells and how T cells work. So the book kind of goes back and forth. Chapters, I think 4 and 5 are sort of this introduction to how B cells and T cells make things like these special receptors on their surface. And then I think chapters 6 and 7 kind of goes back to looking at B cells again and how they actually function in the context of our immune system. And then they go and look at things like T cells again. So there's a little bit of back and forth, but I just follow it because that's the way the book presents it. And I think it kind of makes sense. But you'll see that during this week, this week, we will have your third quiz on the chapter 4 material that we are wrapping up today. So Monday today wraps up chapter 4. So you can look for that quiz later today, and that will be due Thursday night. Next week heads into our first exam. So our first exam will be next Friday. And the exam, I'll talk more about it as we get closer to it. But the exam is taken on Laulima during our normal class session. Okay, so you need to just make sure that you have access to Laulima and can take the exam. Next Wednesday, instead of a new lecture, I will do a review session. So you would log in just like normal for class, but it'll be a review of some of the big topics to make sure that you're sort of on the right track for being ready for the exam next week. Okay. All right. So last time we had 2 more slides that I didn't get to because our last lecture went a little long. I had tagged them onto this lecture, and that was the version of the PDF that I sent out. But then when I was looking at it this morning, I realized that's a lot of stuff to get through. So I decided to drop those 2 slides again. But the content is still important. But what I decided to do was to actually talk about that content by using some of the other slides from today. All right. So hopefully, if you follow today's lecture well, even if you went back and looked at those 2 slides that we didn't get to last time, they'll make sense because it has to do with what we're talking today. We were talking about the production of immunoglobulins. Remember, immunoglobulins are these Y-shaped molecules that have the 2 arms that are the antigen binding domains. These are the arms that have the 3-dimensional shapes on the ends that are unique for binding some 3-dimensional shape. And that shape that they bind to, the antigen, might be a molecule that is present as part of a pathogen or maybe is made by a pathogen, right? And so the key here is to remember that this is part of your adaptive immune system, because if our innate immune system was already good enough and successful at being able to detect that pathogen because of things like our complement protein being able to bind to it easily, or macrophages, neutrophils being able to eat up and destroy the pathogen, well, then we wouldn't even need to engage our adaptive immune response, right? So what we're talking about now is, since our innate system failed to take care of the problem, like instead of just normal rainbow-haired clown, we're talking about bad clown, right? Something different that escapes our innate system. We have to rely upon our immunoglobulins to be able to come up with the identification and being able to recognize something about this pathogen that can be used, right? So that our other immune cells can take that knowledge and be able to target it successfully. So that has to do with the immunoglobulins that first start off on the surface of our B cells, because they're made by our B cells, right? Only our B cells make immunoglobulins. And we said, importantly, every single day, your body's making hundreds of millions of different brand new B cells. Each one, its surface is covered with a unique immunoglobulin that it came up with itself. That particular B cell came up with the sequence of amino acids to make this unique immunoglobulin so that it can recognize a particular three-dimensional shape. As just a way of our immune system kind of going overboard and anticipating different shapes that it's never seen before. I always say our immune system is not psychic. It doesn't know what bad clown is going to look like if bad clown comes along a week from now, a month from now, a year from now. So you have to have some immunoglobulins that just by chance are the close enough fit to be able to bind to different features, three-dimensional features on molecules from bad clown. All right, so our B cells do this, and the way we get such incredible diversity of shapes is through this VDJ recombination. So recall that typical immunoglobulin is this Y-shaped molecule. It's made up of two heavy chains. These are the long chains in the middle here, 1, 2, and then 2 light chains, 3, 4. So that's 4 chains total. Right? And these light chains are made by 2 different genetic loci in our bodies. On chromosome 22, we have the lambda light chain locus. Locus just means it's a collection along our DNA on chromosome 22 of different fragments that can be pieced together to make a complete gene, to make a lambda light chain, right? So you have 2 copies of that because you have the chromosome 22 that came from mom, you have the chromosome 22 that came from dad. Likewise, on chromosome 2, you have the kappa light chain. Again, it's a group of gene fragments that can be put together, picking and choosing, to make a kappa light chain. And then on chromosome 14, you have the heavy chain locus, right? A whole bunch of these fragments, including now these what are shown as green D blocks. Right? And remember, each one of these, especially the variable domains, the red ones here, there are many to choose from. So that's why the cartoon draws it this way. It puts little dotted lines, little breaks in here to remind you that these things are not necessarily super close to each other. They can be quite spaced apart on this long segment of your chromosome. And it puts brackets around these to show, well, here's the first V block, here's the second one, and then there's another, like 30 or so over here, right? So this is representing it as by putting these brackets around there. And then somewhere pretty far downstream, there's a little break here, far downstream, you have your J blocks and then your constant blocks. Okay. So recall that we had been comparing this to an analogy of putting together a cookie recipe, right? So I always like thinking about our genetic information as being sets of instructions that are like recipes for making protein, right? Or in this case, recipe for making a cookie, right? So I think this concept is kind of hard to imagine, though, of how these different blocks can come together. So let's say, if we look at, for example, the recipe or for a white chocolate McNut cookie, if you think back to the story that I was saying last time, let's say there's a cookie contest and some students, they presented their version of the white chocolate McNut cookie recipe, right? So their recipes might vary in terms of certain ingredients, right? Not necessarily completely 100 miles apart from each other, because they're all still kind of limited to having to make a white chocolate chip McNut cookie, right, of a particular size and shape as far as the cookie contest. But their choices of different things that go into their recipe is what's going to result in slight variations of the finished product. Right, the prize-winning cookie, and maybe the second or third place runner-up as well. So that's what we're talking about here. different pieces of information come together to make slight variations of the antigen binding sites in the immunoglobulin. So if we think about it this way, last time I used a long piece of rope to represent the chromosome. And along the chromosome, let us say that this is chromosome 14, for example. This is the region of the rope that contains all the fragments for making immunoglobulin heavy chain, right? Or IgH for heavy chain. So there's around 40 or so red blocks to choose from. There are, like 23 or so green blocks to choose from, 6 yellow blocks, and then maybe 9 different constant blue blocks to choose from. You can think of these as just being sort of different groups of ingredients. So you have, these would be the flour that you choose, and there might be like 40 different brands of flour, right? So if you're an avid baker, maybe there's a particular brand of flour that you really like to use for your cookies. Different brands of sugar that would have slightly different properties, right? Some of them are very finely powdered, some of them are more coarse, right? So your choice of which sugar to use would have a subtle but important variation on the recipe. And then similarly, mac nuts, and then you have a more limited number of shapes that you can choose. So we said you have this protein complex, the recombinase activation complex, so the RAG protein, like this oven mitt, that is able to randomly choose a red block, a green block, and a J block to put together. Right? So these are just fragments, right? And so remember, the way that the oven mitt works is that it has part of it combined to those highly conserved 9 DNA sequence, 9-mer sequences, the purple sequences, that are spaced apart by either 12 or 23 bases of DNA from a highly conserved 7-mer, right? 7 DNA bases that are highly conserved. And this particular spacing of this side having 12 spaces between, or this side having 23, that's the only way the oven mitt can successfully grab onto it. That's these RSS, or recombinase flanking sequences, signal sequences, are what help the selection of the colored blocks to make sense, so that the blocks are not lined up backwards when this loop of rope is brought together. So in this case, it randomly joined this green block and this yellow block, right? And anything in between, like any of these green blocks, yellow blocks that were in between, are just lost, right? Because the rope is going to get cut here, and then the 2 frayed loose ends of the rope have to be joined together. Right? And if that happens over here too, then you end up getting a choice of which V block was chosen, which D block, and which J block, and then eventually it'll go ahead and add on a C block. So that's like choosing this brand of sugar, this brand of flour, this brand of sugar, and this brand of mac nuts, along with this shape, right? And then that's how you end up with your particular white chocolate mac nut recipe. Okay. So a couple of you asked me, how this actually corresponds to these hypervariable loops in the ends of these variable domains of the immunoglobulins, right? So at the ends of the Y-shaped molecule, the ends are the variable parts. So that's what this VDJ recombination is meant to contribute to. And I said, if we took the variable domain of the light chain over here, right, and remember that most of it is yellow, meaning most of it is pretty standard and it doesn't vary from immunoglobulin to immunoglobulin molecule that you randomly sample from your blood, let's say. But in these red regions, you see very high variability in the amino acid sequence, right? And that's what gives then the overall 3-dimensional shape of the right part of this, variation, right? And that's what's going to determine what this thing can fit up against, right? Just like 2 puzzle pieces coming together. If you look at this diagram over here, you'll see that these hypervariable regions, remember, this is if you actually took this globular protein and sort of laid it out, stretched it out. Most of it are these yellow parts that are not very variable from immunoglobulin to immunoglobulin. But when you get into these red regions, where you have a lot of variability in the amino acid sequence, you can see that it turns out that the HV1 and HV2 loop, right? So that's these 2 loops over here. those are simply determined by which V block gets chosen. So you have, let's say, 30 or so different V blocks to choose from. They are more or less going to be highly similar to each other with some variations in the DNA sequence here, right? That makes V block number 1 different than V block number 15 or V block number 30, right? So the choice of which V block already gives you what the 2 hypervariable domains are that you are starting with. The third loop here, the hypervariable domain number 3 over here, this is a little confusing, right? This is actually a function of the J block that's going to be chosen to go with this V block and the way that random DNA bases are used here to join the ends of the rope that have been cut. Because remember, that whole loop of rope that was in between these two, once the oven mitt decided to jump over a whole bunch of stuff and choose this red block and this yellow J block, right? All that stuff is lost and the rope is cut. The DNA is cut. So it has to be joined up together again. And the way our cells do it is by randomly adding DNA bases to each cut end until there's enough of an overlap that the 2 strands can then stick together again. And then you have DNA polymerases that fill in the rest, right? So that short little stretch of DNA bases is brand new to your genome, right? Because it was just randomly put together to get these 2 ends of the DNA to stick together. So that's the third hypervariable loop. So that's where these 3 hypervariable loops come from, so that when the resulting, all these fragments are put together, the V, the J, maybe a D block, and then that is then transcribed by RNA polymerase, and you make RNA that gets processed into mRNA and then translated from RNA into the finished polypeptide sequence, which is the big, what you see here, the ribbon diagram of the amino acid sequence, right? That's where these 3 blocks come from. So it's a combination then. You have the choice of the blocks themselves, gives you a lot of variation, but then how they're joined together randomly by pieces of DNA, that's a lot of variation, right? And then on top of that, this is just to make one chain, like the light chain, right? This is also happening to make the heavy chain. So then the combination of all the possible light chains and all the possible heavy chains, right? That's what really blows the variation out of the water, right? So you can make 10 to the 12, roughly speaking, different shapes of immunoglobulin antigen binding domains. Okay. So here's another way of thinking about it. These 3 things are going to come together here: the V block, the D block, the J block. The hypervariable loops 1 and 2 just come from whichever V block you chose. Hypervariable region 3 actually comes from these random DNA bases that are used to reattach the cut DNA between the D and the J and the D and the V. The reason that sounds confusing is this makes it look like there'd be 2 regions of variability. In reality, this D block is super short, right? It's only something like 3 or 4 codons of DNA. So the cartoon kind of makes it a little, hard to imagine. But in reality, once this is joined together, this is the third hypervariable loop, right? And then when this resulting gene made up of these separate fragments are chosen and put together, and it's transcribed and translated, that's where you get your heavy chain, right? Because then the variable regions, so together the red, green, and yellow together make the variable heavy chain region. So that's now this red part here. Right? And then you will add on a choice of the C blocks to make the stem. Right? So that's what makes this particular recipe for an immunoglobulin or white chocolate McNut cookie unique, right? Okay. So this is another illustration that hopefully helps you kind of visualize this, right? In the light chain only involves VJ recombination because there's no green D blocks in the lambda or kappa light chain gene loci, right? So just as long as you choose one V block and one J block, okay, and one C block, right? So this is now the rearranged DNA. Right, so instead of there being a whole bunch of red blocks here that are not shown and several yellow blocks, now you've made the choice. The oven mitt has successfully put together this here. So this is rearranged DNA then, right? Then the primary RNA transcript that gets transcribed from this would have these components in it, the V, the J, and the C block, right? It would then undergo splicing to go ahead and get rid of this extra intronic region of RNA that's not needed. So that puts then the variable domain close or right up against the constant domain. And then this finished mRNA would get translated in the cytoplasm of your B cell by the ribosome to make the finished polypeptide chain, right? And that's why now they're showing, oh yeah, that's a reminder that the variable domain is actually a combination in the light chain of the V block and the J block. Okay. The heavy chain, similar story, except for now you have this additional D block, right? But same thing. It gets chosen, rearranged, and so you get now this VDJ recombined over here. And then the eventual edited mRNA, right, also has the heavy chain constant regions. Okay. So I can understand if this VDJ is very confusing. I think I mentioned to you last time, I never learned this as an undergraduate. None of my undergrad molecular cell bio classes at UC Berkeley taught this at the time. I encountered this in graduate school. And so I actually remember when I was learning this in grad school, you know, I could not fathom this at all. This made no sense to me at all. The professor who taught it was a very esteemed, well-respected cancer biologist. He actually was the guy who discovered one of the important proteins that's involved in unraveling DNA as part of DNA replication. So anyhow, he had his lab group, and the lab group that I belonged with, you know, we were on the same floor, and I used to see him all the time. He was a very nice guy. You know, you'd always be like, "Hey, hi Howard, how's it going?" I'm like, "Oh, hi Mike," you know, and he was always really nice to me. And then, you know, I took the class where he was one of the faculty members, and he taught this VDJ recombination stuff, right? And I was completely lost. And so when I took the exam, I like bombed his part of the test, right? I completely like, you know, and so I was really embarrassed because I was like, oh, he's going to hate me now because somehow I'm like not worthy because I don't understand the stuff that he actually, you know, does research on. And so one day afterwards, I actually went up to him like, hey, Mike, I'm really sorry I bombed that part of your test, you know. And I was kind of like, I think I was kind of stupid about it. I'm like, you know, yeah, but I really like it. It's interesting. But anyhow, it was really funny. I thought he'd be all mad at me, but he was like, yeah, I don't really care, right? Because he's like, yeah, I know you're a smart guy and it's fine, whatever. And I was like, oh, okay. So the truth is, you guys are learning this as part of an undergraduate class. That's well before I was ever exposed to it. I think most college students don't have this. So if it's difficult, you know, don't be discouraged, right? Like I told you, I didn't get it either. But that doesn't mean it's like totally okay for you to bomb it on the exam, right? I'm not going to get mad at you, but I'm not also telling you the story just because I'm telling you it's okay for you not to understand it, because this is actually really important for our understanding of how it is that our body makes antibodies. Okay. So there are, again, 5 different isotypes or 5 classes of immunoglobulins. And your B cells will produce these depending upon what's needed at the time. As we learn more about how our adaptive immune system deals with different types of infection, because maybe suffice it to say right now that a bacterial infection is very different than a viral infection. It's very different than a parasitic infection. Right? So these different isotypes of immunoglobulin, due to slight variations in their structure, are better suited for dealing with different types of pathogens. A single B cell, once it's come up with its unique cookie recipe to make this particular immunoglobulin on its surface, remember the important part is the red part. That's what the B cell came up with through combining V, D, and J, right? So that's the unique recipe that this B cell has. The rest of it, the stem, the blue parts of these, that can kind of be swapped around, right? So it's depending upon which blue parts you have here is what determines the 5 isotypes of immunoglobulin. But the important thing to keep in mind is the red part, the variable part, right? That's the thing the B cell needs to hang on to and not change radically, right? Because that's the whole reason this particular B cell turns out to be valuable is because by chance, its recipe for immunoglobulin makes the right shape to be able to bind to something on the surface of bad clown. So as a B cell switches between making these different isotypes, IgG, M, D, A, and E, right? The red part is the part that it kind of retains. And that's the part of the recipe that it's not going to mess with a lot, right? It's just going to change, for example, the blue part. So in the cookie analogy, I said the blue part is kind of like choosing the different little, what do you call it, mold that you can finally, once you make your cookie dough and everything, right? You can either have a heart shape, star shape, moon shape, whatever. I think you can imagine that part, right, does change what the cookie looks like at the end, right? Just like changing the blue parts of these stems would change the properties of the immunoglobulin. But really, what you're not changing is the actual recipe that goes into the cookie, right? Because that's the important part that you want to hang on to, right? So each one of these has both a membrane-bound form, which we call the B cell receptor form, and a form that's no longer anchored to the membrane, that's free to float away. And as a review, that's what an antibody is, right? The free-floating soluble form of an immunoglobulin made by a B cell. All right. So the first Igs that are made by a brand new B cell that's coming out of your bone marrow, right? One of hundreds of millions today, right? So this little B cell, right, it's practically indistinguishable from all of its brothers and sisters, B cells coming out of the bone marrow, right? And the vast majority of them, guess what, are not going to ever be useful for anything, right? Because the little three-dimensional shaped immunoglobulin ends that they've come up with turn out to be, not useful, right? They're not going to bind anything, right? So the very first immunoglobulin that any B cell makes is the IgM form. So here you see the IgM. So it means, let's say that this B cell makes a VDJ recombination choices of the different blocks to make the variable domain. Then it also chooses the constant mu navy blue blocks, right? And there's this extra stuff at the end. You'll see as part of these mu blocks, there's this orange group here and this light blue group of gene fragments here. So the primary transcript RNA that comes off of this has everything on there, right? The edited mRNA, right, has all the introns cut out. So the blocks are put together now in one continuous sequence. And you can see here, it's cut out the orange part, right? And instead it has left the light blue part. So when this mRNA gets translated into the finished amino acid sequence, the polypeptide chain for the heavy chain, right, that light blue part codes for the little piece of peptide at the end, the peptide sequence, that's the anchor, right? So it's this hydrophobic region that keeps this Y-shaped receptor anchored to the cell membrane, right? Later, if it turns out that this B cell is actually chosen because its immunoglobulin ends up binding something like a molecule on bad clown, right? So suddenly it becomes valuable, right? It's no longer just one of 100 million or so, B cells floating around. This one's actually selected as being one that works or something. It may be told to make the free-floating antibody form of this IgM. So what happens then? Here, it still has the same VDJ choices that it made, right? Because that's the part that's actually working to actually bind something. And for most of the C blocks, it's going to be the same. But now look, as part of the RNA editing, right, the little light blue parts are discarded and the orange part is kept. And this orange part is just the end of the stem that no longer anchors it to the surface of the cell. Right, so this form of the Ig now can float away from the cell, right? So this is the secreted IgM form. In other words, this is the antibody form of this IgM. Okay. So all B cells start off by making IgM and some IgD, right? So those are 2 out of the 5 classes of Ig that all B cells start off making. They make IgM and IgD at the same time. And that's the only example when a B cell can make two different isotypes at the same time. The IgD, it's not shown on this figure, but it's actually on one of those from last last lecture that I decided not to include today. So this is what I'm saying. You can go back and look at it, and hopefully this will make sense. The C delta blocks also end up being part of the primary transcript RNA. All right, so you'd end up with not only all of the C mu blocks as shown here, and sort of the ends over here of the C mu blocks, right? But this would actually keep going, and you'd also get a whole bunch of the C delta blocks as part of it. Right? Then, as part of mRNA editing, some of them will go the way that this cartoon shows and get made into the C mu constant peptide sequence to make IgM. Other molecules would end up having this stuff all cut out, and they would keep the C sub delta blocks. And so you'd end up making an IgD molecule. All right. So those two features, being able to switch between the membrane anchored form versus the free floating form, in other words, between B cell receptor form and antibody form, and being able to switch between production of IgM and IgD, and why one cell can make both IgM and D at the same time, those have to just do with how you edit the RNA. Right? Other class switching between, let's say, IgG or A or E, is a more fundamental involved process that starts at the level of DNA. In other words, in those cases, you would not have the C sub mu or C sub D blocks ever be part of the primary RNA transcript, right? The actual assembly of the complete genetic sequence up here would involve choosing the C sub alpha blocks or the C sub G blocks or the C sub epsilon blocks, so that those would be part of the RNA message. So that's why a B cell cannot switch. Once a B cell switches from one to the other and becomes a plasma cell, right? The plasma cell that's making antibodies can no longer go back and switch between isotypes. Right? So the IgM represents the first B cell receptor that's made and the first form of antibody that gets released. So remember, the IgM, your cell came up with it just by that whole sort of random process of VDJ recombination. The fact that it actually successfully binds something, like the two close enough fit puzzle pieces in the box of puzzle pieces that I shook up, hey, that's purely by chance, right? But it's lucky chance, because now you have the start of something that you can use, right, to work on, to make detection of the pathogen better. Right? So IgM then, even though it's the first antibody that's made, because when a B cell is chosen, right, up until that point, your body doesn't have any antibodies against bad clowns. So now all of a sudden you have this one B cell that knows how to make something that can bind to bad clown. Yeah, you better believe your body's going to want that B cell to start making the antibody form of this, right? So through different RNA editing, it makes the free floating form of its IgM, right? So this IgM is kind of like the first quick and dirty antibody response, right? Because we're still dealing with close enough fit puzzle pieces here. It's not great, but at least it's something, right? So the IgM, here it is as a monomer by itself, so one molecule. So if you have 5 molecules of these and a particular protein, that's called the J chain protein. I know it's confusing because this is not the same J as the little yellow J blocks in the VDJ recombination. This is a different protein, but that protein kind of acts as a little clip, and it clips together 5 of these IgMs in a pentameric ring, right? And you can see here, that's the formation of this ring here. So now this pentameric ring has 10 antigen binding sites on it. So this is a very good way of attaching to something like the surface of a pathogen, right? If these antigen binding domains recognize a certain thing on the pathogen. But remember, each one of these is a pretty weak binding, right? Because it was just chosen based upon close enough fit, the 2 random puzzle pieces that came together. So IgM is good for neutralizing or blocking certain antigens, right? But it's not very good at recruiting things like other immune effector cells. Why? Because remember, The stem part of the Y-shaped molecule is the part that usually attracts other cells, right? In a pentameric IgM ring like this, those stems are all kind of hidden, right? But what it is really good at is it can bind and cover the surface of a pathogen, and it can recruit complement proteins, right? So this is one way our adaptive system helps recruit complement to the surface of a bacteria where the complement before was not having a good time, right? So this is then part of the third complement activation pathway that we studied previously, the classical pathway, right? We had studied it because we said that C-reactive protein that's made by the liver is made to help complement protein stick. In this case, what we're saying is if your body previously, right, did not have any exposure to this thing, but now your body has made these IgMs that also recognize the surface of the bacteria. Now these pentameric IgM rings can also help complement protein stick. Okay. So the main limitation here, though, again, is that you're dealing with the original shape that was come up randomly through VDJ recombination made by this B cell, right, that you only now realize, you know, is valuable because there's a close enough fit between it and a surface feature of the pathogen. So clearly, though, right, we need to improve the fit, right? Now that we've chosen like a puzzle piece that is close enough to bind to the other puzzle piece here, right? We don't want just close enough. What we really need is to figure out how to make that fit as good as it can be, because the stronger the binding will be, right, the more efficient your immune system can target and deal with this pathogen. So how does this B cell know how to improve the fit of its immunoglobulin? Right. So this is a truly remarkable kind of phenomenon. And that kind of has to do with this, right? So this researcher here, right, this is Professor Charles Xavier. He's a very renowned geneticist, human geneticist, right? And so this is a quote from him. He says, "Mutations, it's the key to our evolution. It has enabled us to evolve from a single-celled organism into the dominant species on the planet. This process is slow and normally takes thousands and thousands of years, but every few hundred millennia, evolution leaps forward," right? So some of you, might recognize Professor Xavier. You're like, I don't know, I saw him somewhere, or maybe it was like a Nova special. No. So here he is with his colleagues. Okay, so this is his research group, and all of them are doing a lot of cutting edge work with mutations and human genomics. So as part of his group, here's his undergraduate researcher who's very bright-eyed and eager and excited even to have some quality undergraduate lab experience, right? She has a great future ahead of her. Over here is the jaded, overworked, and underpaid and underappreciated postdoc, right? Whose career is not going anywhere fast. But anyhow, so I think many of you have seen the X-Men movies, right? And maybe you've seen them with your friends. You go to the theater, and then afterwards, your friends who are not, biology majors or not life science majors, they might be like, whoa, that's crazy. The idea that all of a sudden things can mutate and we can have these like crazy mutations. And maybe they ask you, is that even possible? Right? And you're a life science major. You're like, no, mutations don't work that way, right? Mutations do take hundreds of thousands of years, natural selection, blah, 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 right? I mean, you even remember like I think the opening credits of the movie, they're like zooming around through, I don't know what this is, DNA or something. And then all of a sudden there's like this little explosion, right? And it's meant to be a mutation where something is altered. So, I'm going to kind of blow your mind here, but it turns out that somatic hypermutation is real, right? And your body, your B cells have been doing this ever since you were born. Right? So what is somatic hypermutation? It literally is super accelerated, punctuated natural selection. We call it hypermutation. Hypermutation that takes place in these immunoglobulin gene loci, right, that make the light chain and heavy chain. So the way it works is that you have an enzyme, or I should say more specifically, your B cells express an enzyme that goes around and randomly deaminates or modifies cytidine residues in your DNA. Okay? So this enzyme is called activation-induced cytidine deaminase, and it results in anywhere you have a C base in your DNA, it replaces it. It basically deaminates it so that this nucleotide, this base needs to be removed, and any 4 of the bases are put in its place. It could be a C again, in which case there is no mutation, but it could also be an A or T or a G. So this happens as a B cell is dividing and making 2 daughter cells. There will be these random mutations that are introduced in the VDJ gene fragments that B cell has. Keep in mind, this B cell that's dividing already is a B cell that successfully came up with its own VDJ recombination and came up with its own cookie recipe. And that's why it was released from the bone marrow and it went to work. And now it was identified that its particular recipe for immunoglobulins gave a close enough fit, right, to attach to the pathogen. What we're saying is this B cell now is told, in order to try to improve or change the fit, it's going to undergo random mutations at its immunoglobulin site, right? So when it splits into daughter cells, those daughter cells then have slightly different Ig loci. The genetic sequence that encodes for the VDJ block that's part of the chosen recipe have now been altered. Right? If you look here, this is an interesting bit of clinical data that highlights this. So this is looking at patients after they're immunized, right? They're given a vaccine to start their immune system selecting for B cells that know how to make particular immunoglobulins that maybe bind to the primary targets in the vaccine. Right? So what they're looking at here is in a population of B cells, they're looking at the immunoglobulin M, right? And there's 2 sets of genes, right? The ones that come together to form the variable region of the heavy chain, and then the V region of the light chain. And remember, each one of these has these 3 variable loops that determine the shape of the binding. So previously in the slides, we've called these HV1, HV2, and HV3 for highly variable or hypervariable regions. The book is a little sloppy because here they start calling it complementary determining region, but that's what it is, right? It's a loop that determines what this thing is complementary to. Right? So what you see is 1 week after being immunized, if you take a sample of different B cells from the person's blood, and you look at the 3 hypervariable regions that are part of the Ig recipe. So this is what, 1, 2, 3, 4, 5, 6, 7, 8 different cells, right? And you look at the VDJ recipe that it has inside for making immunoglobulins. What you see is after 1 week, you see random mutations happening in these hypervariable regions, right? In the heavy chain and also in the light chain. Okay. If you look 2 weeks afterwards in the same patients and take another set of, let's say, 8 B cells, look at how widespread the mutations are beginning to be, right? So very clearly, in a short amount of time, just a couple weeks, right, you've undergone several mutations of the immunoglobulin domains in these cells, right? So that is pretty amazing. We are talking about something like 200,000 years worth of mutations happening in 2 weeks, right? Because it's natural selection. Because the cells that have these mutations, most of them, right, as the cell is dividing and the daughter cells carry these new mutations, you can imagine that most of them screw things up, right? If you're just talking about randomly substituting DNA bases in there, the shape of the immunoglobulin antigen binding domain, if anything, there's a greater risk that it's not going to bind as well as the original version, right? So yeah, there are many of those that just get discarded because they don't work any better, but a small subset of them, by chance, the random mutations will make the fit of the immunoglobulin with the antigen tighter, right? So those daughter cells, those B cell daughter cells that have tighter fit get preferentially chosen now, right? Some of them are told, hey, your immunoglobulins are better than what we started with. You start making antibody forms of those because that's better than the antibodies that we had a few days ago, right? Other of those daughter cells are told, you keep doing the hypermutation, right? So you're getting better and better and better waves of B cells with better and better immunoglobulins just through this hypermutation process. Okay. Now I know what you're saying. You're saying like, that's not like X-Men, because X-Men's all about like mutations that let you shoot like lasers out of your eyes, or you can walk through walls, or like control the weather or something like that. This is kind of disappointing, right? No, it's not, right? Because just think about this. Like in the X-Men movies, they have these different what is it? These different skills that help, protect them against things, right? Like making a force field around them or something like that. Think about it, right? This is your body rearranging its own genome to fight against an enemy that you had never previously seen before, right? I mean, that is exactly what this is. And you're doing it by doing this extremely accelerated evolutionary process. Right? Again, 200 years worth of evolution crammed into 2 weeks to save your life, right? That, I think, is pretty awesome. Okay, so here is a table that helps you begin to understand why you have different immunoglobulin subclasses. Right? I said these all vary by differences in what stem region the B cell decides to put onto its variable region that it came up with. Right? You can see that here are the 5 subtypes. There's IgM, IgD. There's also IgG. So there's actually subclasses of IgG, and then A, and then E. Right? So the reality is M, D, G, A, and E. Yeah, that's the 5 subtypes. This table shows you that because of their variations in shape, because of the stem region, they actually have different properties that make them useful for certain things more than others. For example, IgG, which is the most common antibody form that you find floating in your blood, like about 70% of the immunoglobulins that you have floating in your blood right now are the IgG form. They're very good at neutralizing, meaning they're good at binding to the surface of viruses and bacteria, preventing them from, let's say, infecting cells, right? But maybe they're not as good, for example, at doing something like activating the complement system, whereas IgM and IgD are very good at activating the complement proteins and getting complement proteins to stick to the surface, right? But they may not be as good at doing things like, for example, neutralizing, right, or opsonizing, right? As we go through the semester, we will look at these different subclasses in more depth as we understand or begin to understand how our immune system has to differently deal with different types of infections. Right? This last slide, then, I'm not going to go through this because this is just a kind of recap of what we were talking about today. I encourage you, as part of your studying, to look at this and understand it. Again, I don't want you to memorize it because that's not my point in trying to teach you something, right? But I want you to be able to read this and think about what it says, right? Because what this is talking about is as a B cell is forming in your bone marrow and it starts doing the VDJ recombination to come up with its own unique Ig recipe, right? There are certain consequences, meaning at certain steps along the way, it has rearranged the genome in it in a way that's irreversible, right? Or there are some steps that can go back and forth because it doesn't involve genomic rearrangement. It has to do with alternative RNA editing and splicing, right? So look at these, try to make sense of it. I think they phrase this as sort of like changes in the genes of a B cell throughout its life. Okay, this will become even more clear to you as we discuss this further. One thing I did want to end with, though, is all this talk about somatic hypermutation, right? And comparing it to X-Men, something like that. The one thing that you do have to keep in mind right, is those characters in X-Men, the implication is every cell in their body has this mutation, right, that lets them shoot laser beams or control the weather or whatever, right? In the case of your B cells, it's only individual B cells, right, that are each going through these steps that come up with certain changes in their genome, right? So in other words, only a subset of cells in your body, the B cells, would have slightly different genomes based upon this hypermutation thing. The rest of your body, right, the rest of your tissues, right, they still very much just have the genome that you were born with. So keep that in mind, especially because, think about it. What does that mean in terms of these variations of B cells actually being something that you can pass on to your kids, right? That'll become very important later on when we talk about this idea of how infants are protected using mom's immune system, right? Okay, but that's for a future discussion. So keep an eye out for the quiz. I'll make that available so that you can take it by Thursday night. And I hope you guys have a good rest of your day. I'm going to go ahead and stop the recording, and I'm happy to answer any questions that you might have. Okay, let me stop the recording here.